We are live.
Namaskar. Welcome to the 130th edition of Gyan Pravah, the podcast series by Foxy, which is the brainchild of our president, Dr. Sunita Tandulwadkar, ma'am. I would like to introduce ma'am in brief. Right now, she is in Europe in EBCon, and as usual, is mesmerizing everyone present over there. She is the HOD IVF and Endoscopy Center, Ruby Hall Clinic, Pune, founder and medical director of Solo Clinic IVF, a center for excellence in infertility at Pune, and co-founder and medical director of Alive, a stem cell research and application center, Pune. She has the extraordinary achievement of having the India's first stem cell baby and world's first at the age of 45 on 16th April 2018. She is currently our president. She is President Foxy 2025, President-elect ISAR 24 to 26, Founder President Society of Ovarian Rejuvenation, President POGS 2122, Chairperson, Founder, Honorary Secretary ISAR, President IAG 2019-20, VP West Zone Foxy 2015 to 17, elected board member of ISGE 2013 to 17. She is the reviewer and advisor, Journal of Human Reproductive Sciences, editor-in-chief, Indian Journal of Gynec Endoscopy. She has authored 18 books, 85 publications. She's an invited faculty to 400 plus national and international conferences. Her award list is very, very long, but there are a few that can be mentioned. She has the IVF Icon of the Year 2022, Pride of Foxy 2020, Bharat Gaurav Puraskar 2019, Best Medical Practice 2016 from the European Medical Association, Best Citizen of India Award 2016, Glory of India Award 2016, Pune Shlok Ahilyabai Holkar Award 2015, Rashtriya Gaurav Award 2015. She has a letter of appreciation from the President of India in 2012, Sarvshri Award 2012. In her stead, we have our most dynamic Joint Secretary, Dr. Ashwini Kale ma'am, who has been sparing her time endlessly for all our queries throughout the year. So welcome, Dr. Ashwini. Over to you.
Thank you so much, Asha madam. A warm welcome to Vamsi sir. The topic you have chosen today is so different, and this is one of the most dynamic topics where India has made so much of progress in the past 10 years, and there is still so much more to be done. And it is always interesting to listen about genetics. And I just went through Sir's CV and I saw that he's a lot of interest in computer applications as well. So, sir, for your knowledge, Foxy this year for the first time has also developed an app for the patients where they can get information about all the general gynec and obstetric conditions from authentic doctors. All the blogs, all the articles are written by our gynec members, which are about 46,000 across the country. So all these articles are written by our 30 committee chairpersons. Because these days on social media, you get so much of half-baked information. So we thought, why not spare authentic information with them? So in collaboration with UNICEF, this year in the month of January, we launched this app. So it is really doing very well. And even the ASHA workers find it very handy to use it because there are small general questions as what the anemia level should be. When should I fit in the copper T? When should I remove it? So it is very useful for them. We also have launched an app on contraception, which is an AI-based app. So basically there, if you just put in the information by any layman, they come to know what are their options of contraception. Because we see these days in the general OPD, everybody is so busy in the OPD that there's hardly any time left for counseling these patients and telling them their options. So here the patient gets multiple options according to their medical conditions, age, previous history. So it becomes very easy for the patient also to come and ask about a specific contraception because these days we have a huge basket of contraception. And then another project which we are doing under the leadership of Dr. Sunita madam this year is on preconception care. We are stressing a lot on preconception care because very little has been done for this. There is a lot of work which is done for maternal mortality and morbidity once the patient gets pregnant, but nothing before that. So we are trying to optimize our hormonal levels, do a genetic screening, do a thalassemia screening for them, we have a huge project for thalassemia eradication, which is called as the Mukta project. So like these, we are doing many projects for 360-degree for women's healthcare. And this time we are targeting even girls who are more than 12 years of age, so that no stone is unturned. And for the boys, this year, for the first time, we have started a Pehel initiative where we are doing this sexual education in schools, which are specifically boys only school. Because till date, we were only educating our girls and women regarding this. We were targeting only schools and colleges where girls were there. Now we are taking these in co-ed schools, and especially for the boys, we are also educating them regarding the menstrual health and sanitation also, so that somewhere they'll go back and educate their mother, sister, or their wives. So I would now join in from the viewers link, and a very warm welcome once again, sir. It will be lovely to have you with us, and I will listen to this podcast from the viewers link. All the very best, Asha madam. You are muted, madam.
Thank you so much for those kind words. Today, I was in the Aparajita program in Raipur. And as you said rightly, there was so much about preconception, how to take care of a woman, how to correct her micronutrient levels, which translates into future better health. So it was great. So we again have Sunita ma'am to thank for that. And thank you so much.
Thank you, madam. Thank you, sir.
Thank you.
So now I would like to introduce our guest for the evening, Dr. Vanshi Mohan. He is the Chief Scientific Officer at Strand Life Sciences. He was the Vice President of Bioinformatics before that, Director at Software before that, and basically he has been a research associate at Computational Evolutionary Genomics. These are so many words that, even I have to understand what they mean. He has a PhD in computer science and engineering from Penn State University, MS Computer Science and Engineering from Iowa State University. And one beautiful thing is that his PhD was on genomics. So, sir, would you like to just say something about it? What exactly is genomics? This is from a baseline view of a normal gynecologist. Even I had to read up so much to talk to you.
First of all, I'd like to thank you all so much for inviting me for this podcast. It's indeed an honor when I was hearing about all the achievements that you mentioned about your chairperson and for Dr. Ashwini Kale, all the different programs that you're doing. It's indeed an honor to be speaking to this community. So thank you all very much for that.
You're welcome.
Yeah. So as you mentioned, my background really was in computer science. So when I was doing my PhD, that is when, if you recall, in the late 1990s, the human genome sequence was getting assembled. So,
Yes.
all of us know that there are 22 autosomal chromosomes, and then we have the sex chromosomes. But what is the sequence of those chromosomes, the ACGT nucleotide sequence? That is really what was being constructed in the late 1990s. So obviously, each of us will have a different copy of the genome. All of our sequences will be different. So they had to make something called a reference genome. So they pulled together 7 individuals' genomes and they made the reference genome. And that was a $2.7 billion project just to make one genome sequence for them to construct it. That was happening then. And
What's the need? See, I'm a layman. I'm asking you, what was the need? Why did you need to make that genome, that
yeah.
single genome?
So I think when we look around and we see some people have a disease, some people don't have a disease, and then sometimes you see that a disease runs in a family. So you say, what have you inherited from your parents, or why is this disease present more in this particular community where there's consanguineous marriage happening? Which parts of the and the answer would lie in some material that you've inherited. So, what aspect of the genome is causing the disease is what people wanted to know. And once you figure out which portion of the genome is causing the disease, then you have a way of testing whether somebody has that mutation or not. If people are going to marry, you can test whether two people who are marrying, they carry the same copy of the mutation or not. So
My question was very different. What I said was, why did you need to pool 7
So I think they didn't want it to be a specific individual's genome that was getting assembled because all of that was government money. So they just together parts of different individuals and they were all anonymized. And that is what they used to make the reference genome
But
sequence.
then what I'm saying is, supposing while they are, taking different parts, so
Mhm.
some parts may get lost, right?
Not really. They did a lot of overlapping things. They said this portion of the genome from here, and they did a lot of mix and match of things. So I think there would never have been a perfect reference genome for everybody to compare with anyway, because all of us are so different from each other. So you just needed to make some representative thing that you called a reference genome so that everybody can compare with respect to it.
Okay. So, there are tools for genomic analysis. Can
Mhm.
you tell me what are the tools that are used for genomic analysis?
So let us say we take one particular individual and you want to find out what is lurking in their DNA. So first thing you do is you take a blood sample, you'll extract the DNA from it. Then you can't unfortunately sequence the entire chromosome in one go. So you'll chop it up into small pieces. So you chop the DNA into small pieces, then you feed it into the sequencer, and the sequencer will spit out the genomic sequence of all of this. So if you were to sequence a typical individual, you'll end up with roughly around 120 gigabases of data to represent the entire genome from the sequencing. So then you need to compare whatever you got with that reference genome and say what is different between that reference genome and your genome.
So the
So
reference that you took was completely healthy?
no, that reference is just everybody has to compare with something. It's almost like a map. So everybody decides that I'm going to compare with this map. Whether it is correct or not, I'm just going to use
These,
that as a
these
baseline.
questions would be very foolish to you, but they are the questions that, come to my mind, that
Yeah.
you say that this one is different, so this is not normal. So was the primary one totally normal? That is my,
No, nobody would be completely normal.
yeah.
So when we compare against that reference genome, each one of us would have roughly around 4 million differences with respect to the genome. So most of those differences would be completely healthy. They don't cause any change at all. It's just the way that the different populations have evolved, different changes have happened. All of us have acquired different polymorphisms. Some of them would be very common in the Indian population. Some of them might be more common in the Karnataka population. So a lot of these changes might be completely harmless, but a few of those changes might actually be deleterious.
Okay. So how many have we been able to, pinpoint that these are actually not good so far?
Yeah, so this is a quite a complicated topic. So, for instance, we were looking into some trio analysis, saying, let us say you have a father, mother, and child, and then you compare and say, how much are you inheriting? How many of those look dangerous, and so on. So instead of looking at the entire genome, if you were to look only at the genes actually make up only 1% of the entire genome. The rest of the part of the genome has regulatory regions and other stuff that we don't really understand at all. But the 20,000 genes that we have, those are sprinkled around these 22 chromosomes, and they actually correspond to only 1% of the genome. So,
Okay.
if, so even in that 1% of the genome, any one of us has roughly around 150 mutations, which look like they would be causing problems. Nobody really knows if they do or not, but they look like they would spoil the function of the gene.
Okay. So each one has about 150.
Yeah, on an average, when we looked at maybe around 100 trios that we had done the analysis, it looked like each person on an average would have around 150 mutations, which are affecting a copy of the gene. And purely from the bioinformatics perspective, when we say that this is the protein sequence that the gene is encoding, but because of this mutation, either the gene would not function or it might cause a change in the protein structure. If you look at it from that aspect, around 150 different changes seems to be there. And also, what we found out was that you have de novo mutations. It's not like we are always just inheriting from our parents. Sometimes new
What
things
can you
arise.
define a mutation? What is a mutation?
So, so the technical term, I guess, would be a variant. So when we compare our genome with the reference genome, the differences that we see, we would really call them variants. Some of these variants are present in a large number of individuals. So anything present in more than, let us say, 5% of the population, you'd call them a polymorphism. Anything that is present in a very small percentage of the population and it is causing a deleterious effect, those are typically what people are calling mutations now.
Okay. Okay. So, when we send our reports sometimes for analysis, we get that, variant of unknown significance.
Yeah.
And then there is that, supposing I have sent for mental retardation, that child is having some jerks and I've sent it. And when I get the report, there will be 4 or 5 things that will say that this looks, maybe related to this, maybe related, maybe related. And
Yeah.
then there is a whole list of, you know, unknown significance.
Yeah.
What does it actually mean? What I, as a clinician, what am I getting out of that report?
Yeah. So I think maybe around 10, 15 years back, when people didn't have these nomenclatures, you would just look at the mutations and then it becomes very subjective. Is this mutation really the one that the clinician is looking for? Or is it something that happens to be just present? Like I mentioned, each of us carry 150 different alterations, but mutations. But are those really what the clinician is interested in, or is it something else? So then ACMG came up with very strict criteria saying when can you call a mutation as pathogenic? When can you call it likely pathogenic? When must you label it as variant of uncertain significance? So, if it is causing a truncation to the gene, so the gene is getting, the protein is definitely going to get lost, then it kind of reaches the level of a likely pathogenic. But it reaches pathogenic significance only if there is literature evidence for it, or there are animal studies that have been done in it, or there have been segregation studies done on it. So one of those three things needs to be done before a variant can be labeled as pathogenic. Either you should have shown that in some particular family, this variant was present only in the individuals who had the disease, but it was not present in the other members of the family. Either you do that, or you show that there is a mouse model or a zebrafish model or some other animal model where if you induce this mutation, it shows the equivalent phenotype to the human. But if you remove that mutation, then that phenotype goes away. Either you have to do those kind of
You
studies,
have to show a direct cause and
you
effect.
have to show that. Uh,
Okay. And
Otherwise,
then,
you cannot label it as, unfortunately, many times we find a gene that looks like, this probably is the answer for the clinician, but because of the guidelines, you will have to label it as a variant of uncertain significance.
okay. And then there is such a thing as variable expression. What is this variable expression?
Yeah, so sometimes you'd come across a family or an individual where you think now you have recessive disorders and dominant disorders, right? So in dominant disorders, even one copy of the gene, if it is mutated, it can cause the disease. In recessive, only if both the copies of the gene are affected, then it causes the disease. But in dominant conditions, sometimes you'll have two individuals who have a mutation in the gene, but one of them has the disease, one of them does not have the disease. So these incomplete penetrance and variable expression, nobody really knows how they happen. Some people say that um the gene is encoding for a particular protein, but the protein is not working in isolation. The protein interacts with many other proteins. So if maybe some other effect is happening in some other protein, then this mutation in the protein is having a much larger effect. Whereas if the other proteins are all right, maybe it has a lower effect. So nobody fully understands those combinations and permutations, because of which it leads to these.
Means the reason I'm asking you is I have one patient who
Hmm.
had all her tests were normal. The double marker, NIPT, NT scan, this, that. Third trimester, she developed growth retardation, severe growth retardation. And when the baby was born, we tested, it turned out trisomy 21. No cardiac disorder, no shortening of her limbs, no hyperechogenic focus in the heart totally normal, seen by cardiologists. And now that kid is 3 months old,
Hmm.
holding up her head, responding. So she has trisomy 21, there is no doubt about it.
Hmm. Hmm.
But, doing well.
Yeah.
And then there are others who would not recognize their parents even up to the age of 6 months.
Hmm.
So is it possible, supposing that patient comes to you for an MTP, that I don't want to keep this pregnancy, it has trisomy detected. Then how do we as clinicians decide on the expression? Because maybe that child will turn out totally normal.
Yeah.
How is it decided? How is that expression decided? I'm talking to you from a clinical point of view. I send the report to you. How
Hmm.
do you decide the expression?
Yeah, I think right now, unfortunately, from the DNA perspective, we cannot check for the expression at all. So it is possible that if you look at the protein expressions themselves, because the DNA is only serving as a blueprint for the genes to get expressed, for the proteins to get created. So it is possible that if you look into more of the proteomics analysis or the gene expression analysis, maybe it will give a particular clue saying that even though the DNA looks like it is like this, at the protein level, things are actually all right. But unfortunately, I don't have too much experience on that proteomics angle of this.
Okay, but is it happening anywhere in the world? And will it actually come within the reach of patients?
So what I've heard
Because
is,
for that, the funds, the amount that they have to spend also is important. No?
yeah, yeah,
So,
there is some work going on in RNA and DNA.
hmm,
So for instance, not in this particular context, but when you mentioned about variants of uncertain significance, right? So let us say we find a gene and it looks like it has been knocked out. There is a loss of function of that particular gene. But if you want to verify it, then you can say that if it is indeed the gene is lost, there's only one copy of the gene left, which means the amount of RNA that the gene is generating must be lesser than if both the copies had been intact. So there are some research groups which are also trying to go a little bit further on the VUSS by looking into the RNA element of it. But the protein and what you mentioned, that part I haven't heard.
okay, so that's, for you, for the future.
Yeah, yeah.
So now, where are these studies that you do, the genomics?
Mhm.
What is its clinical use? Where is it used?
So there are really two different areas in which we look into genomics. One is on the reproductive health side, and one is in oncology. So in the rare disease and reproductive health, we are largely dealing with the DNA that we are born with and saying what mutations have we inherited and what are their implications. And this can be done in right from NIPS, where you're looking into trisomy 21 and 18, as you mentioned, that is preconception. And immediately after baby is born, if you find any neurological disorders, if you find any musculoskeletal disorders, you want to know what has happened. Again, that is a germline test. Once they grow up, if there are again some developmental delay, if there are epilepsy conditions, again those things. And later on, when people eventually want to marry and they're looking into carrier screening, you could again use germline things. So these are all the different tests that you can use where you're analyzing the DNA that we're born with.
Okay, so in preconception,
Mhm.
a young couple comes to you. When would you advise them to go for carrier studies? What are the specific things that we are confirmed? Like Dr. Ashwini mentioned, thalassemia.
Yeah.
Our area has thalassemia, sickle cell, and a combination of the two. That
Yeah.
is very prevalent in Chhattisgarh. What
Mhm.
else? Hemophilia?
Yes, hemophilia. So we've been offering carrier screening for a long time, but unfortunately, what happens is almost all the cases that we get are usually after they have a child has been born and the child passed away, or a second child is born with again the similar defect. That is when couples really wonder, is there something in the combination of our two genomes that is causing this issue? So a lot of the requests that we get for carrier screening are actually after the fact. We don't really get too many of them preemptively. We get a very small subset. And when we get that subset, as I mentioned, because there are so many genes which have loss of function mutations, as I mentioned, you get 150 different things, right? But many of those are in genes which are not really clinically important. So each person would get around 5 to 10 in the 2000 genes that we shortlist for carrier screening. So now essentially an individual would have 5 to 10 genes. Their partner again would have carrier status in maybe 5 to 10 genes. And then you can compare both of those reports and see are there any genes that are affected in common or not. And that happens relatively rarely, uh, but that is how it goes. But mhm,
My question is that the couple comes to you.
mhm.
Where would I, as a clinician, offer them this study? What are the known disorders other than, sickle cell and thalassemia and
Yeah, so you do have the range of disorders is so large. After the first three or four, then it just becomes a lot of very, very small percentage disorders, unfortunately.
so
So
what are the most common?
most of them are musculoskeletal disorders. We see those. Then we see certain cardiac issues show up prominently in carrier screening. I mean, there is hypertrophic cardiomyopathy, all of those, but those are not really relevant at a young age anyway. Those are for much more older. Uh, then we see metabolic disorders, protein metabolism, uric acid metabolism. Those metabolic disorders, I think, are the ones to pay more attention to. Because if you do find those disorders, then intervention is very easy. The moment the child is there, you can change the diet, you can give supplements, and those can be handled quite effectively.
Okay. So how many rare Mendelian disorders are there?
Yeah, that list seems to grow by the day. Right now, it stands around 7,500, 7,500 or 8,000 rare Mendelian disorders are there, where even with a single mutation and the gene is knocked out, there's a very high chance of getting that particular disease.
My God. And what about how many of them are common? Like out of those 8,000, how many are common?
Common is very few. It's probably less than 10, the names that you mentioned and all. Those are probably the common ones. Because by the very definition of a rare disease, it is, each one of them individually occurs less than 1 in 1,000 in the population. It's just that so many of those diseases are there that overall 5% of the individuals they say are affected by rare diseases.
Okay. Now, one more thing, these days epigenetics is coming into play. There
Mhm.
is so much of a role of preconception and in conception environment of the fetus that causes changes which are not actually transmitted, epigenetic changes. What
Mhm.
is your take on epigenetic changes? Do they change the genome?
Epigenetics is a very, very difficult area. So, as you know, all of the tissues, for instance,
Actually,
Mhm.
as usual, people oversimplify. If there is so much of exposure to carbon, there is smoke, if there is exposure to pollutant, if there is exposure to plastic, the genes are going to change. It's that's how people tell. You take
Mm.
so much of fast food, your child gets programmed to that. I've heard a lot of things, and now with the advent of YouTube.
Mm.
So are these things epigenetics? Does it change the genome? Can you actually measure those changes that are epigenetic changes by
So,
looking at the genome?
yeah, so first, one thing we need to clarify is that epigenetics is something happening on top of the genome. So, you have this ACGT sequence, which is your DNA. And then, when you have CPGs, where you have a C and immediately next to it, you have a G, sometimes there is a methyl group attached to the C, and sometimes there is no methyl group attached to the C. So, that is what we mean by methylation. So, if there is a lot of methylation happening in some particular location in the genome, then that part of the genome is somehow silenced a little bit because all of these different methylation groups are sitting on the DNA, which means that proteins which come to that location and want to cause expression of those genes, they're hindered by this methylation in some sense. So, methylation is really used as a surrogate for silencing of some particular part of the genome. And this is very important because all of our tissues have exactly the same DNA, but different genes are working in different tissues in our body. The heart cells are behaving differently, the skin cells are behaving differently, but all of them have exactly the same DNA. And the way that happens is through methylation. So different parts of the genome are methylated in heart cells, because of which some genes are expressed. Different parts of the genome are methylated in lung cells, because of which different genes are expressed. So methylation is a natural process and a way in which it controls that different genes are expressed in different parts of the body. But sometimes this methylation goes awry. So if the methylation goes off kilter, then suddenly genes that should not get expressed are getting expressed, or things which should be expressed are not getting expressed, and that can cause changes. It cannot cause DNA level changes, but it can cause changes in the expression and the protein levels and have a clinical impact.
Okay. These days, we see a lot of autism.
Mhm.
Originally it used to be like 1 in 3,000. Now it is so much more.
Yeah.
Different studies give different figures, but that rate has increased by leaps and bounds. In
Yeah.
one study, I read up to 30%, which I think is too much, but this is US figure, not Indian.
Mhm.
So I'm just asking you, do we have something specific for autism spectrum disorders?
So this is tricky, because every case that we get, it comes with a list of clinical indications that the doctor writes, and it is almost never just autism spectrum disorder. So sometimes it will be autism spectrum disorder along with epilepsy and along with something else that we see. So there are always these different combinations of indications that come. Sometimes we get just standalone autism spectrum, but that is relatively rare. So
Not just with you, anywhere in the world. Do you have a specific set that this set is responsible for autism?
yeah, so when you put all of these keywords together and say what are the genes involved in these neuronal activity processes or developmental process and so on, so the number of genes that people analyze for autism varies somewhere between 400 to 800 genes. Okay, and and Dr. Harsh Sheth's group in Ahmedabad, actually, the Freech group, they have done some good work on autism. They've done some trios. So now autism, again, it shows up in two manners. One is simplex autism, where only one child is having autism, but the siblings do not have any autism. It is just an individual case. And in those cases, a lot of the times it is de novo variants. It is nothing that the parents had. It is something new that was there only in this particular individual.
We cannot make out why it happened, right?
No, we cannot make out why it happened. There have been papers over the last 10 years which say that de novo variants increase depending on the paternal age at the time of conception. But there are some studies which dispute that. So the evidence is a little bit mixed out there. So in the case of simplex autism, where only one child has it, two-thirds of the cases, I think, there are de novo variants involved. But sometimes you see multiple siblings having autism, and in that case, it's not de novo, because de novo is so rare, it's not going to happen in multiple members of that family. There, it is usually an interplay of multiple genetic factors, each of which is contributing a little bit. So I think autism spectrum disorders really are at the midpoint between these completely Mendelian disorders, where a single mutation is going to cause a disease. And then you have, on the other extreme, you have conditions like diabetes and all that, where you have thousands of genes and thousands of polymorphisms, which slightly increase your risk. So autism is somewhere there. People believe that there are multiple genes and multiple variants which together cause that autism, especially in multiplex cases.
Okay, now I'm taking you from what you said right now, that there are multiple genes that predispose you to diabetes.
Mhm.
Supposing, this predictive genomic thing is also coming up. There are labs that offer you that we can tell you your risk of cancer, we can
Yeah.
tell you your risk of lifestyle disorders.
Mhm.
Get your genomic kundli. That is what, that's the ad.
Mhm.
So how correct is that? And supposing your genes tell you that you're going to get diabetes,
Mhm.
is there anything that you can do to prevent it? Because if you cannot do anything to prevent it, what's the point of it?
Yeah, you're absolutely right. Um, so I think there's a whole range of these diseases, right? For instance, when you look into hereditary breast and ovarian cancer, for instance, there, if you have mutations in the BRCA genes, the odds of getting cancer increase sixfold. So there, I would say that you're looking at a very small set of genes and you're looking at a high odds ratio. So that is one category of tests. But when you look at diabetes and other things, you're looking at these have typically come through genome-wide association studies where they look into 6,000 locations in the genome. They analyze all of those using some models, and the risk is only going up by 30% or so. It's not like a dramatic increase in odds ratio. And many of these models have been developed in the West. And how well do they translate into the Indian context? What are the largest studies to do that? Um, yeah, I'm not very strong proponent of this polygenic risk scoring things. I'm not an expert on that, but you need very large studies in the Indian context before you were to take a strong stance on those.
As things stand today, we should not be trusting blindly on these kundlis.
Yeah,
So that is the take home basically.
that is absolutely my point of view. Yes.
So now we come to the infertility panel. Our president ma'am is a infertility specialist. You saw Ashwini. Many people in Gynae are now focusing on infertility. Do you have something specially that can predict that this couple or this lady or this man are going to have infertility? Are there markers to mark infertility?
We have been working on a study which is funded by BIRAC on idiopathic infertility. So the last 3 years we've been looking into because infertility is a well-studied topic. Doctors already have a lot of different clues on which they act. So we wanted to focus a little bit more on idiopathic infertility and see will we find new genetic markers which haven't made an appearance so far. And so far we have sequenced 700 women clinically diagnosed with idiopathic infertility. And as controls, we also sequenced 200 mothers who are willing to be part of the project to act as controls. And so far, we come across around 200 genes where we see loss of function mutations only in the probands and not having any loss of function in any of the mothers. Among them, around 100 of them look like candidates where we didn't find these mutations even in global databases of women who had conceived and so on. Now, these genes go all across the spectrum. Now, we're trying to go through the literature and say, where do these 106 genes or 200 genes, into which parts of the overall reproductive cycle do they fall? All the way starting from follicle development to oocyte maturation, to the actual fertilization process, to the transport of it, to the embryo development, and so on. We came across a couple of them. For instance, we had two instances of PAT-L2 gene, and this gene is needed for oocyte maturation. And if this gene is mutated, then the oocytes remain in the GV phase. They do not progress to the M1 and M2 phase. And this was something we saw in two probands out of 700. And when we went through the literature, just 3 years ago, also other groups had also published on PATL2. They had mouse models for it, and it is indeed a strong candidate. So like that, I think doing the sequencing and coming up with the hypothesis is only the first part, because until you do animal studies, until you do other literature things, it becomes harder. So one interesting thing we found is we found a whole lot of um, 10 to 15 probands where we found mutations in the dynein family of genes. So these are the genes which help in the cilia movement, and usually they've been implicated in male infertility because sperm will not be able to navigate up the canal, etc. But there is emerging evidence that they're also involved in female infertility. So, for instance, the cilia movement that is needed for the oocyte to move, or the cilia movement that is also supporting the sperm to move upstream, those might get affected in this family of genes. So many of these are fairly early findings. We'll have to work with some clinicians working on infertility, or we'll have to do more focused
So if
studies.
the cilia in the women are not working, are these the women who repeatedly get ectopic pregnancies?
So that's a great question. Unfortunately, when we designed the study, we didn't have access to any of the clinical reports. We had only the blood samples.
Means
So
I'm
now,
just thinking, because
yeah,
if somebody is getting repeated ectopic pregnancies
yes,
and has patent tubes, so this cilia thing could be one of the
it
reasons,
could
right?
be. It could absolutely be. So we need to see how to work with more clinicians in the field and researchers to see, these are the patterns that we're seeing. Are they supported by what you see in the clinical records or not? Yeah.
Okay. So one thing I wanted to ask you, as a clinician, can I order a gene test for infertility?
So we have around 400 genes that we have been studying. So I think you can order the test, but what we would need is a open conversation saying that we are seeing these mutations in the gene based on our understanding of literature. This is what the genes do. Does it correspond with the phenotype
So
that you
it's not
see?
commercially available right now.
It's not commercially available right now.
Okay. So that's one thing. Even the ovum maturation thing, not available.
Yeah, that is something we found out only the last few days, but that very specific use case, it could be used, but I'm not sure you'd want to order with such a low frequency event, right? You'd probably want to cover a lot of events if you're ordering a test.
Now, there is one more thing. These days, we see a lot of premature ovarian failure,
Mhm.
a lot of it. Young girls are coming up as young as 16, 18 who are getting ovarian failure. Is there some gene panel which we can order for premature ovarian failure? Because these girls, they come with, having regular periods for 2, 3 years, and then they start having irregular periods. So
Mhm.
if we have a panel which we can order at that time, we can at least save their ovum, ovum can be saved.
Yeah, unfortunately, right now we don't. We're
You
hoping
don't
that
have a panel for premature ovarian failure?
no, in the next 6 months, we hope to consolidate all of these findings and offer something. But at the very moment, we don't have something.
Okay, but it will be there in 6 months, right?
Yes, in 6 months, we will.
Okay. So I think NIPT is something gynecologists have been discussing day in and day out. But there is one question that I have about NIPT as well. When would you consider it a valid test? One,
Mhm.
because yesterday only one company came and said that we are validated for a fetal fraction of 2%,
Mhm.
right? We give results and they are valid. So when do you consider it valid? What fetal fraction? And which report would you consider? What is the detection of, trisomy 21, 18, 13, XY? And then these days, they give all 46
Mhm.
chromosomes.
Yeah.
So how valid are those reports?
So when you look at NIPT, the method for detecting NIPT itself from a computational perspective, it's actually very straightforward. It's because you're sequencing all of the DNA, you're extracting cell-free DNA from the blood, and a certain part of the cell-free DNA is coming from the fetus, most of it is coming from the mother. When you sequence all of it and you're looking at all of the chromosomes, you're saying how much of the data is coming from each chromosome? Am I getting a little bit extra from one chromosome? If I'm getting a little bit extra, maybe it is coming from the fetus, right? So that is
Fetal fraction.
that is the underlying assumption. Now you could be getting a little bit extra because of irregularities in the way the mother's DNA was processed. So many other things also could cause it. So the higher the fetal fraction that you have, the more easier it becomes to see the signal, saying that I'm definitely getting more in this chromosome. If you're going down on that fetal fraction, then even a subtle change in that thing, can you really trust it? Is it noise? It becomes challenging. So one way to overcome this is by doing a lot of sequencing. If instead of doing, let us say, 10 million reads, you're doing 20 million or 50 million reads, if you sequence at a very, very high level, then even the subtle changes you can make out. So I would say that
But
if,
is that mentioned in the report that this many levels have been done?
yeah, so I think those are the questions you would need to ask, saying that,
Because I'm a clinician, I read the report.
yeah,
But what are the intricacies I need to know to trust that report?
yeah.
That is a very basic question that I have.
Yeah. So
What fetal fraction would you say is acceptable?
I would say that you should, most people do it at 4% fetal fraction. That is when I think the results look solid. But anybody else who comes to you, what I would say is, ask for two parameters. Ask for how much sequencing are you doing, and also ask them to give a confidence level for that measurement, because you can detect maybe a certain chromosome looks like it's getting more data, but is it statistically significant or not, and try to get a confidence level for it. And that one people usually do using Z scores and all, saying how many standard deviations away. Anything more than 3 is trustworthy, usually, because that is 99% confident. So I think those are the, unfortunately,
They are not
clinicians,
mentioned in the report.
they're not mentioned. And as you get into more and more of the subtle signals, unless the statistics is something
The
clinicians
reason I'm
look at.
asking you is because it's somebody's life.
Yeah.
It's somebody's life that we are dealing with. You just say this looks abnormal and the patient goes there and has an abortion. So it's
Hmm.
life that you are dealing with. How reliable are those reports? So you look for, on a baseline, 4% fetal
4%
fraction,
limit.
and you look for a z-score of more than 3,
3. Yeah.
plus minus 3.
Yeah.
Okay. So we are done with that. Now we come to the last part, and that is the cancer. Okay. You spoke about methylation studies.
Mhm.
We understood a bit about that. Now, how confident are we that we can predict cancer?
Yeah. So we've been
Two
working.
things: predict cancer and predict the personalized treatment for cancer.
Okay. So I think I briefly talked about hereditary cancer and BRCA, so let's not get back into that. So suppose somebody has a familial risk of cancer, or they have an occupation because of which they think they're at higher risk of cancer, or they're a heavy smoker, they want to know if they have cancer or not. So we really want to catch cancer at the early stages. And so many groups are working on early cancer detection, and we have also been working on it for the last 5 years. So the basic principle there is that every part of every tissue in the body sheds some amount of DNA into the bloodstream. That is the whole basis for even NIPS testing. The fetus is shedding some amount of DNA, and we're trying to compare the fetal DNA versus the maternal DNA. Similarly, if you have cancer, the cancer tissue is also dividing, and the cancer will also shed some amount of DNA into the bloodstream. So when you collect cell-free DNA from the blood, a lot of it is coming from leukocytes and regular cells, but a small fraction would also come from cancer if there was cancer. So now how do you distinguish this cancer cell-free DNA from regular DNA is the main question. Now, there are two ways you can do it. Either you can look for mutations, which is what we use when you're doing a treatment, for instance. For instance, lung cancer, we would look for EGFR mutations. For colon cancer, we look for KRAS mutations. So you could either look for mutations, but mutations are very, very few. So you might catch it, you might not catch it, because in the early stages of cancer, there is so little of cell-free DNA in the bloodstream. Or you could look for methylation signals, which are much more wider. So when cancer is happening, in the cancer cells, there is a whole scale change in methylation patterns. There's a lot of methylation that goes away, and there are many areas where the methylation just shoots up quite a bit. So these dramatic changes are happening all across the cancer cells. So even if there's only a small amount of the DNA floating around in the bloodstream, by looking at methylation patterns, you have a decent chance of capturing it. So that is the premise.
Would it be able to tell you which type of cancer it is? Or just that it is cancer? Means, you
Yeah.
get what I'm saying?
Yes, yes. That's a great question, because once you find out it is a cancer, what do you do next, right? Because there's only a screening test. You'll still have to go to a doctor and say, I might have cancer. And all of the current screening diagnosis methods are focused on particular cancers. If it's a lung cancer, the doctor knows to do a low dose CT. If it's a breast cancer, they can do a mammogram. If you just go and say cancer, what are they supposed to do? So
So would the person directly go for a PET CT?
I
That
think,
would also.
yeah, yeah. So, yeah, these are all emerging technologies. So, we've looked into two types of signals. One is it cancer, yes or no? That turns out to be relatively easy to detect. And with 80% sensitivity and 97% specificity, we can do that. But the question you asked afterwards, that is the tough one. Say, can you predict which cancer it is? And there we are able to predict only 75% of the time. We are able to give two cancers and say it might be one of these two cancers. Now, this we understand is a dilemma. So we're saying that it could be cancer, and we're saying that it could be one of these two cancers. What do you do next, right? So this is the conversation that we're having with a lot of oncologists around the country saying, suppose we were to do this report, is it at all useful for you? Would you rather we restrict all of our analysis only to the screenable cancers? Because we can detect 10 cancers in this manner. But let us say that we detect a cancer which you don't have a screening method for, then what are you supposed to do? Should we, should we restrict only to four cancers which are screenable? Should we do it all? And we're hearing a whole lot of different opinions. Some of them are very enthusiastic and say, I will guide the patient, you just let me know and I'll manage it. Some people say, no, I want to be very, very focused only on these specific cancers. I cannot handle anything else, it will cause anxiety. So I think development of technology is one thing, but translating it into practice where we work with clinicians is
Which
a tough.
cancer is something that you are not very sure of right now. But supposing you've done your methylation study, you know there is a cancer. Can you even predict which drug would work on it? Because then you could take the drug and the cancer would go away, right?
Yeah, so in fact, that is exactly what one of the oncologists said. Suppose we were to do low doses of some particular drug and it goes away and see the change in signal, then maybe that is the right one. But these are all very, very hypothetical situations, and there's no way that a testing company like us would even go down that path. So we
I'm
just.
not talking just about your company. I'm
Mhm.
talking to you. You have done your PhD. You would be reading research. Is
Yeah.
there anything anywhere in the world that actually tells you that this drug would work better? This drug would work better on this.
Mhm.
Personalized cancer care. Is
Yeah.
it anywhere on the horizon? And is it effective?
Personal
Because you
cancer.
can do it and it doesn't really help, and you've extended life by spending maybe 50 lakhs for 2 weeks. It doesn't work.
Yeah. So personalized cancer works quite well when you're looking at mutations because that is where most of the therapies have been developed. So this methylation analysis is not going to give you any insight at all on personalized treatment. For that, we do have to find the cancer. We need to take the biopsy. We need to find the mutations, and only that can help with.
So you found a mutation. How does that decide which treatment to give?
Yeah. So I think. So there are all of this FDA information about for each, there are around 50 genes where if you have mutations in them, there are targeted therapies which specifically target that particular gene. So in lung cancer, for instance, the EGFR mutations, they make up around, I think, 60 to 70% of lung cancer cases. There are very precise drugs that target those. Now, the efficacy of those is variable. Some of them, as you mentioned, only extend lifespan by a few months. Some of them extended by a couple of years. So the efficacy is variable, but 70% of lung cancer cases are actionable. In breast cancer, again, 50% of breast cancer cases have personalized medicines which are FDA approved. And there are another 20 to
But
30%
they
of
don't
them.
give you life more than a year.
Breast cancer, I think some of the PIK3CA things and all give you a couple of years.
That's what.
Yeah.
They don't give you more than that. See, my younger sister was diagnosed directly stage 4 with
Hmm.
nothing in her breast. It was her thoracic spine biopsy that told us what it is. It was a metastatic breast cancer, which is a separate category.
Yeah. Yeah.
And then she was on Palsib. It worked well for a year and
Mhm.
3 months, and then it stopped working.
Yeah. So the only things which seem to have a profound effect are immunotherapies. So immunotherapies are hit and a miss. When they work, they seem to work extremely well, and there have been some very long-term survivals. But predicting who will respond to immunotherapy and who will not, people haven't
Can your
come.
genes do it? Can genomics do it?
So right now, the eligibility for immunotherapy is coming through genomics. So you can rule out a lot of people who are absolutely not going to respond. That part of it seems to fine. And then you say that the tumor mutation burden is high, so there is a chance that immunotherapy could work. But amongst those, only around 40% of them seem to actually respond. So there are lots of efforts going on people trying to find out of those people who should ideally be responding. Can we still figure out who will respond and who will not respond? There's a lot of research going on that area, but it hasn't really gone through.
There is light at the end of the tunnel.
Yeah.
There is light.
Yes.
So I think I've taken up a lot of your time. Is there something you would like to say at the end, which would be a take home for all of us?
Um, I think there's a lot of great initiatives that all of you are doing. That's what I heard earlier on. The whole many of us are researchers. We work on reading literature, doing experiments, and so on. But the whole reason we work on this is to eventually have clinical translational benefits to it. And we come up with a lot of hypotheses. We would like to engage with doctors. If the doctors are able to keep good medical records so that we can correlate genotypes and phenotypes, that is what we see as the biggest challenge. Sometimes we find some particular genotype, but the doctors don't have the records. They may not have made some observations because they're so busy dealing with the patient care. So we'll just
I
have to
think you need to design a form.
yeah.
You design a form that, even the doctor's assistant can fill up. If there is a case that is going for genomic study, then, keep this data. And if
Yeah.
that form is filled, then there's no issue.
Yeah. Yes.
So I think you need to do that
They
work.
need to do that work. Yes.
And then we will see that we as clinicians document better. I think, every time there is a conversation around doctors, it boils down to documentation. Document better.
Yeah.
And we as Foxians, if there is any part of your study where you think we could help, I think Madam would be very glad to have Foxy involved in all of it.
Yeah.
Thank you so much. We have come a long way, but we have a longer way to go.
Yes.
Thank you so much for your time.
Thank you so
Namaskar.
much. Have a good evening.
Good day.